Got it.
Okay. So yesterday when I left off on the protein activity, the DNA to protein thing, I gave you the last part and I said it was on Edmodo and I wanted you to complete it. But I felt like there wasn't enough information, so I thought what I'll do is really quick. I'll make sure that we do a little bit of review. I think most of you guys got it, especially when we get today when you read there's a codon chart that you guys did yesterday. It took a little bit of time for everybody to do it, but you got it. It wasn't that hard. It just took a little bit of time to figure out, well, how'd they get from here and how'd they get to here. I'll give you some of the specifics because the sheet that I gave you yesterday, I did post a study guide on there, and I think it's fairly complete. I went through it, and I haven't checked it with a test, but if you've got everything from the DNA replication number 2 worksheet, and you've got that sheet from yesterday, I'm not going to overload you with a lot of redundant questions. It's just that the stuff that I want you to get is going to be, everything we're doing is just like the basic stuff. We're not doing anything more than that. I think your biggest challenge is just going to be making sure you get the mitosis and the parts of the mitosis, and the one more thing that we're going to do today, okay, which is just basically, yesterday you went from DNA to amino acid to protein. We're going to go backwards today. We're going to go, here's a trait, here's the amino acid to code for the trait, let's go all the way back to DNA. All right? So I want to give you some of the specific information, but I wanted to use this diagram, and so you can use this sheet as notes. There's a backside, it's totally blank as well. You can use that for notes. There's one more activity that's on the back of that we'll go through, and we'll just, hey, this is a day for practice. Let's make sure we're good to go with all this, and then we'll see where it takes us. Okay? So I wanted you to kind of see this. You have the nucleus right here, and I want to make sure that you have this. The two things. So we've talked about replication and transcription translation. So let's do this. Replication. It's kind of hard to visualize some of this stuff, so I'm just trying to give you diagrams and such. Replications and transcription. And the best way to think about transcription is your copying. Okay? Those two things happen in the nucleus. All right? So I'm going to take a little arrow. And takes the right enzymes, a little bit of ATP. We're going to unzip just like we did with replication. And you're only going to copy one side, all right? So this is your transcription thing. Put a little star next to that, and then I'll put a 1. We're going to transcribe.
What's the stop for?
Putting a star next to the transcription because we're going to do this step by step. So in fact, I'll do one more thing. Right underneath this, I'm going to talk about, I should have labeled this and said, hey, we're doing protein synthesis. Which is making proteins from DNA code through transcription / translation.
Wait. Code or DNA code?
From the DNA code.
Oh.
I don't have other colored markers.
Down there.
Right down there?
So is the sole purpose of RNA to make protein?
Yeah. Well, to make copies of the DNA to send out. We'll talk about that a little bit more. Here's the other thing too. Once the RNA is read, I can break it all down again. I can break it all down again, and I can reuse the nucleotides and make another RNA message. And then once I'm done reading that, I can break it down and make another one. Okay. So let's look at transcription real quick. Transcription, basically what that means to copy DNA. Just like a lot of other things, you have to have the right enzyme and a little bit of ATP. We'll open DNA at specific spot. When we open it at the specific spot as well, the free nucleotides that joined in and did the replication process, you're going to end up having the same thing. So you have free RNA nucleotides. Attach / copy, and they're not really copying, they're doing the complement of it, so it inadvertently does the copying. But we're only going to do one side of DNA.
So these are all things that happen during transcription.
Yeah, these are all things that happen during transcription, correct. Once we have the strand made, you can see what, we got this? Once we have the strand copied, this strand can now leave, and you guys saw in the video yesterday, the nuclear pores. That's the pathway for information to go back and forth through the nucleus. The RNA strands will leave the nucleus, go through the nuclear pore, go out in the cytoplasm, and it will travel along the endoplasmic reticulum until it hits a ribosome. Now the ribosome looks huge on here compared to what we saw before because it's blown up more. But that's essentially what's going to happen. The ribosome's going to grab it, and that's where that protein synthesis starts happening. So we'll do this. Translation. What happens during the translation process, and then we'll do this. This is your, so part 1, part 2, 1 and 2. We're going to transcribe first, then we're going to translate. Translation happens right here. And this is kind of like, whereas transcribing is copying, translating is kind of like reading. And reading the mRNA, messenger RNA. So what ends up happening is the mRNA leaves nucleus. Ribosomes read each codon to attach. Here, I'll bring it up. Leaves nucleus. Ribosomes. Is that right? Ribosomes read each codon to attach. I'm going to do this. AA is amino acid, okay? To attach amino acids. And then what happens is amino acids join into chains that make what?
Protein.
Protein's good. We're good with that? I'm going to flip to the back page of this. Ready?
Not ready.
10 more seconds. This is one of the other reasons I decided to do this, because it didn't have the information in the packet. Okay? There's 26 amino acids that are used in organic things. We only use 20 of those different ones. Okay? So our whole life, that's on top of the ATGC, which have these basic nucleotides. You also, those code for one of 20 amino acids. And I always think of it like this. The amino acids are like Legos. And if you take the right Legos and you stack them the right way, you can make something completely different than if you take another set of Legos and you build them another way. Okay? So it kind of depends on the type of Legos you're using, how many of the Legos you're using, and what sequence you're using. Okay? So I'll give you this little brief stuff on the proteins and the amino acids really quick. Like I said, I thought this would be better and a little bit more useful to you. Okay, proteins. Let's do this. Proteins made.
So you do it on the same side of the next.
You can do it. You can do it whatever you want. I just have to go to the back side of this.
proteins made what?
That's 3.
I'm gonna do proteins made. So humans, let's see, made from amino acids. No, it's not 3. 1 and 2 refer to the 2 parts of the protein synthesis process. Okay. So now we're just going to what are proteins. Yep. Yep. Let me get that up a little bit.
So this just refers to what
Just
the
the
proteins
basics
are.
of what are proteins. Yep,
Okay.
we'll do that. So made from amino acids. 20 different AA used by humans. So we only use 20 of them. And actually that codon chart that you guys looked at yesterday, there's 20 amino acids on there, but you saw there's 64 different possible combinations. And you'll see one other thing that how that kind of works later on with regard to like mutations and changes in your DNA and how that would actually be effective.
So what are the other ones for?
They're just ones that we don't use. They're ones that other living things do use them for whatever reason. I don't know evolutionarily why they determined, like why different organisms use them and we don't use them. We just use 20 of them, but there are 26 different ones that other different living organisms use. Those other 6. I don't know who uses them or.
Like for different functions and stuff?
Yeah, they just make different types of proteins. That's it. Yeah,
Do
Alex.
you have those in us, we just don't use them? Or
We
is it
don't.
like-
We probably do get them in us from the foods that we eat, but we don't use them. We don't use them. And anything that's nitrogen based, like amino acids, that's why they talk about if you have a high protein diet and stuff like that. If you're taking in too many proteins and stuff, and your body's not using them, you actually have to filter them out. And that's one of those things that's kind of hard on your kidneys to filter out those amino acids and stuff like that. Your body doesn't actually use them. The ones that we don't use at all, that your body just filters out and gets rid of them. So we only use those 20 different ones. And here's how these proteins differ. Proteins differ in these 3 things. You can put a star next to this. This is you just got to know this. Not only do you have to know this for the test, but this is another 1 of 65 in the course assessment. And we just did it earlier today. They differ in number. All proteins differ in the number of amino acids, the kind of amino acids, and you can say order or sequence. Yeah, sorry, of amino acids. Thank you, Lana.
Like kind of amino acids, you mean like changing a yellow to a red?
Yeah, but do you notice that there were lots of yellows on there?
Yes.
Okay, that referred to groups of amino acids. So when you get into it a little bit more today, you'll see that you can switch up with those amino acids. I wanted you to see that because some are what they call hydrophilic, some are hydrophobic, and that's really what does the bending. But that's the other cool thing. I could switch some of those amino acids out, like a yellow for a yellow. Would that make a difference?
No.
No, it wouldn't. That's another way. It's a fail-safe in biological systems of going, I need to make this protein, but I constantly have things under attack trying to mutate and change the code just because it's the nature of where we live. So as long as you have one of these amino acids, you'll still be fine in like a mutational sense. So you'll see that even if you have mutations that don't affect you, we talked about the skin cells here being mutating genes for stomach cells. Well, as long as you have the same type amino acids, sometimes it also won't make a difference and you won't notice, okay? So when we're making proteins made, it's done by the RNA. See, done by RNA and ribosomes. And RNA, I wanted to just do this. It's ribonucleic acid. So I don't know if you can figure this one out. What's different between this and deoxyribose nucleic acid?
There's no sugar in this.
There is a sugar.
Oh, what does that say?
It is a sugar. It's a different type of sugar. Okay, so we'll put that next to it. Different sugar. Now, I would normally call this ribose nucleic acid because of the whole deoxyribose, but it's not. Nobody ever calls it. They just assume that you understand that it's ribose nucleic acid. Just know this. It's a different sugar than the deoxyribose. So it's essentially this. It's a ribose sugar, but Deoxyribose, because it's deoxy, is missing an oxygen. That's all it is. It's not a big deal. All right. So deoxyribose, missing a sugar. DNA and RNA are similar, both code. But differ what's the shape of DNA?
Double
What's the shape of RNA?
double helix.
It's a strand. Strand. It's one strand. I'll squeeze in one strand right there.
Will you go up?
Like that. So we have one strand. Shape is different. And what was the major difference between DNA and RNA? You got- no, the sugar's different, but the major thing you discovered yesterday, A and U, no thymine. Only, what was the other stuff called? Uracil. 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 No, uracil. Okay. All right. So there's no thymine, there's only uracil. The only hard part about that is when you're going back and forth and back and forth and back and forth. You just got to know this. DNA has thymine, RNA doesn't. So if you're complementing and you're going back and forth, so you code, you transcribe a DNA in it. You take an A to a U, but if you go backwards and you got a U and you're going back to DNA, you know that you got to go to an A. You always got to think of the U as basically a T. And sometimes when we actually do it and we're doing the complement of it, I'll say, hey, cross out every T and just basically turn it into a U and flip it the other way. DNA has thymine, RNA doesn't. You'll get used to it. Okay. We done with this one? Okay. Go ahead and go to the next sheet. Go to the next sheet. There's a little practice thing. And I'll basically show you what the deal is with this really quick.
Wait, so we're changing all the T's right here?
No. Don't do that. No. We're not doing that. Only in RNA. So just watch. I'm going to give you a chance to practice this. I'm just going to grab a pencil or something. Yes, it is. All right, so this has got a couple steps. I've never gotten a chance to redo this. I should because I don't like the way it's set up, but I have it, and actually this isn't even a copy. It's just like a PDF that I was able to copy. You'll either see DNA strands in a linear form going horizontally, or it may go vertically. I have one that I'm going to do later, it's vertically. Oftentimes on tests, you'll see it just kind of going horizontally. Based on that, what's the first thing you got to do when you see a DNA strand?
Mark the codons.
Mark your codons. Do it right now. Go all the way across and mark your codons off.
What's
And now, the codon thing kind of makes sense because you're like, oh, now I know why we're doing codons.
What's the importance of codons?
Codons, the DNA is, the RNA reads the codons. It doesn't read single things at a time. It looks at one codon at a time. And a codon, so you guys did the matrix with 64 different combinations. That's how you figure out what amino acid it codes for, okay? So watch, we do this. I'm gonna mark off my codons.
So each amino acid is code for 3.
Each codon codes for a specific amino acid. There's some amino acids are coded by different codons, okay? So watch, I'm gonna show you something. It says look at the DNA strand below, make the RNA strand out of the DNA strand that is marked, all right? Now watch. Number 2 and 3, I want you to do on your own. Where's the RNA strand made? Where does it go from there? Just leave it alone. But number 4 says rewrite the RNA strand and mark the codons. Don't do anything yet. I don't want you to rewrite the whole thing. And then it says below that, write down the correct amino acid sequence. So it's like rewrite. Let's do this. This strand is your DNA, so let's put down DNA. Below that, let's just put the RNA strand below it, okay? And since we're putting the RNA strand below it, you're basically transcribing, well, we'll leave it alone, let's just put RNA strand. What? We'll put RNA strand below it, and for each one of these, we're gonna come, let's do this. I'm gonna extend these a little bit further, so I can have the information below it, okay? So extend yours a little bit lower. Take a moment to do that. Now, I want you to look at the first one, and then I'm going to let you go in a second here. For the first one, RNA, if I'm going to do the complement of it, C goes to what?
G.
G. G goes to what?
C.
A goes to what?
U.
That's where it goes to U. So we're going to go U. Do the next one. What is the next one going to be?
A.
A. See, still A is just no U's. Okay. What's the next one?
A. C. U.
A, C, U. Okay, so you got the idea. Now watch. Below that one, let's go ahead and do the amino acid sequence. So I'm going to put AA below that one. You might have to write it in sideways a little bit. All right. What I've done on this, and this is where I felt like I probably wasted a little bit of paper, I put the codon chart on this one as well as the next one that you have, okay? So go ahead and flip to the codon chart. It's on the back. And we're going to do a little bit of a quiz here. Well, our first RNA sequence is GCU, right? Yes? Okay, so this first column refers to the first letter. The second column is the second letter. The third column is the third letter. So what I do is I go like this. This is how the trick, and I think you guys already figured this out already. G, here's G, that means that our amino acid is in this column right here, okay? We go to the C. C means that we're in this, well, this column, and this is this row, which narrows it down to this group right here. U, we go down to here in this block, we have U. That's the amino acid that we're using. You got it?
You guys didn't get that yesterday?
You got it? Okay, so
I did, but I didn't like it. This part looks really
okay, so now watch. So our amino acid's alanine, right? Yes? Okay, so put it on the other side, alanine.
Do you want us to write alanine or can we write ALA?
It doesn't matter. Alanine. That's why I do it sideways so that you can see. Yeah.
Are we going to know those names of the amino acids?
You should be familiar with them, but they will be available to you.
That'll be,
All this will be
so we'll have
available to you.
that chart.
All, the chart will be available to you. Some of them will be simplified ones. There's one right here. Look,
We're not going to have to memorize
Watch.
this chart.
there's one where I've actually knocked out the 64 different combinations, and I put down here just these. GCU, I just put down the basic ones. And I said, just be able to go back and forth from it. So watch. Go to the back of your codon chart. Let's try this a couple more times. Here's your codon. Get the amino acid in your head. We're going to go C. The codon is CCG. Don't shout it out. We go CCG. Look and find out what the codon, the amino acid is. Five seconds. 3, 2, 1. What's the amino acid? Proline. Proline. Let's do another one. How many got the proline? Okay, Alan, you got it? Okay, let's do GAC. GAC. Three, 2, 1. Seth, did you get it?
Yeah, ASP.
It's aspartic acid. Aspartic acid. Just look it up. Okay. Are we okay with that?
Yeah.
Okay. So here's what I would like you to do. Go ahead and finish up this sheet. When you're done with that, have me check it off. But here, I want you to go to the sheet that you got yesterday from me that has all like the equivalent information. Like half that stuff says, hey, compare and contrast translation transcription. Hey, compare and contrast DNA and RNA. You should be able to fill that out now. That is your study guide stuff, man. I'm just telling you. I got 2 more days. Yeah, that one. Hold it up, Marcel. Turn it. I mean, I think everybody was supposed to get it yesterday. If not, it's on Edmodo.
Yeah, the hard copy.
Okay? Questions? Okay, as soon as you're done with that, check it out. But look, we're moving on today. We've got 2 more things we're going to look at. So if you're not done in time, I'm moving on. If you got extra time, use it wisely. All right? Go.